Hi, my name is David Chagne. I'm from Plant and Food Research in New Zealand. And today I'm going to talk about my work that I do at Plant and Food Research with my colleagues from Genomics Aotearoa about developing genome assemblies for native species of New Zealand. And I'll talk about some indigenous considerations. Before I start my talk, I'd like to acknowledge the sponsor of this session of Plant Genomes 2022. And the sponsor is Horticultural Research, which is an open access journal that publishes some very high quality papers in this plant sector. And this journal is extremely successful at the moment with an excellent impact factor, thanks to the leadership of its editor-in-chief, my friend Max Chagne, who's done a lot of excellent work to raise this journal to this standard to this day. I can't really help you with this slide, but I'll leave it for a few seconds for Chinese speaker to read a few of those words. And I'll just point to some of the activities that Horticultural Research has been doing. Not only it's an open access journal, it also organizes high quality conferences, and it's been doing this since its inception in 2014. The next conference will be held in Wuhan, a beautiful city in the Lake District of China, mid-October in 2022. So please, register and attend if you can. All right, so I'll move now to my presentation, and that's the plan of my talk. I'll be talking mainly in two parts. One part will be about some of our experience about achieving high quality plant genome assemblies. And the second part will be about my perspective and experience working with indigenous communities doing genomics research. Just to be clear, I don't consider myself as an indigenous person. I'm a Gascon from the south of France. This is not an indigenous community, but I'm very proud of my ancestry. But I'm not talking on behalf of indigenous people. I'm really talking on behalf of me as a European born person who does research on the other side of the planet in New Zealand with indigenous people. All right, so as you all know, the DNA sequencing cost in the last few years has drastically decreased. Accuracy and throughput have increased. The new bioinformatics tools coming almost every day. And this means that in the last 15 years, since the first plant genomes, Arabidopsis and rice were sequenced in the early 2000s, it's actually more 22 years than 15 years. There's been a lot of plant genomes being assembled and sequenced. However, a lot of those genomes are not really of very high quality. A lot of them have many gaps, missing data, and are highly fragmented. So how can we produce higher quality genome assemblies? And what I mean by that is high quality, I mean highly contiguous, non-fragmented genome assemblies that are pretty much at chromosome scale. And that means almost no gap or few gaps, low number of missing bases, and possibly like conjugates or scaffolds going from one telomere to the other. Those genomes also need to be complete and cover all the genes and the genomic features, including the complex regions that are notoriously hard to assemble. But also very importantly is that those genomes need to be phased and resolved to their homologous chromosome, not collapsed, not put into a consensus sequence. Also interestingly is that in many of our plant species that we study, often you would have contaminants in your DNA sample that you use for sequencing. So you often end up with some metagenomes, in fact, where it's coming from pathogens or other contaminants, or even endophytic organisms that live in symbiosis with the plant host. You've got to consider also that we're dealing sometimes with more than one species in our DNA experiments. All right, so why do we need high quality genomes? And I'll just show just briefly a couple of examples. High quality genomes, if they've got the specs that I just talked about, being phased, complete, gapless, that allows you to resolve some pretty complex regions that we couldn't address before. Like I'm giving here an example of some of the work that we published about sequencing the bilberry genome. And basically, we looked at some of the loci that are key to bilberry and its close cousin blueberry. And one of those loci is the MIB-A locus, which is critical for anthocyanin synthesis and regulation. And what we knew about the blueberry genome is that there was a few gaps, a few missing data in there and incomplete gene copies. But with better technologies and a more complete assembly of the bilberry genome, we're able to have a gapless assembly around that locus that's got like 5 copies of that particular MIB gene, and we could resolve which one is the most active and the one that's critical for the anthocyanin synthesis. Another example is that once you've got a high quality genome, you've got better chance to do a better read alignments, whether or not you're doing transcriptomic analysis for your gene expression analysis, or if you want to look at detecting variants for either doing GWAS or doing genomic prediction for your breeding program. If you've got a better genome, you've got better variant calls and more accurate, and you may end up often detecting some causative variants for your traits, which drastically improve your prediction if you are doing genomic selection or marker-assisted selection. All right, so in terms of sequencing technologies, there's a few options, but Illumina is quite a popular one. However, the reads are short, even if they're very accurate, very affordable. And those reads, typically, paired reads, of 150 or 250 base pair are not suited to go through complex regions. And we haven't been able to produce high quality genome that are totally complete and gapless using Illumina sequencing. This is particularly a case in plants, like giving an example of maize, for example, that's full of transposable elements, up to 80%. Sometimes, species even bigger than maize, like pine, where there's even more transposable elements, bigger genomes, totally impossible to assemble with short reads. Some other challenges in plants is that there's a lot of polyploid species in plants and a lot of heterozygous species due to either diarchy or due to self-incompatibility loci. So an example here is that, auto tetraploid potato has 1 SNP every 29 base pair, which means it's quite challenging to do a genome assembly for potato when you've got so many variants coming from, different alleles and the diversity of the subgenomes. So obviously, Illumina is extremely useful for many applications, but for scaffold, for assembling a high quality genome, long read sequencing technology are probably the best solution at the moment. And I'll talk about a couple of those options, Oxford Nanopore and Pacbio, and I'll give some examples of what we've been doing. So you must have heard of Oxford Nanopore. The attractiveness of it is that it creates some very long reads and gigabases of data in a run. It's relatively affordable, and they've got a range of different instruments, including some portable ones, like next to my hand on this picture. And the throughput would look like the one I've got in the top, the bottom left part of my slide, where my colleague Elena Lopez-Girona from Plant and Food has been doing some sequencing of an apple rootstock, and that's a sort of distribution of reads that you get, where in this case she's got about 25 kb and 50 reads, which is excellent, and some reads up to 70 kb, which is quite exciting, and that gives you, like enough data, 100x coverage of these to put a really, really good assembly. But I want to talk about this assembly. I will show another example of also of what you can do with Oxford Nanopore is that if you're skilled enough in the lab, you can actually produce some extremely long DNA fragments that you can sequence using this technology. And this is an example here from my colleague Elena Hilario about sequencing some red raspberry. and she generated fragments, greater than 1 megabase, which you can imagine, if you start thinking about putting a de novo assembly or scaffolding your genome, such size of reads is starting to make your life much easier in terms of stitching a genome together. So I'll show some example of work that we've done with Oxford Nanopore, and this is the work that was led by my colleague Anne McCartney. And what we did is that we worked with a native species of New Zealand as part of the Proteaceae family. It's a diploid genome, fortunately, but quite a large one. I mean, largest one for a plant, about gigabases in size. We estimated the heterozygosity between 0.1 and 1%, and we generated some Oxford Nanopore using the Promethean technology, where we obtained about 100x coverage of Oxford Nanopore data with read length about 28 KB. So what we did, instead of doing one assembly using one approach, we actually tested systematically different types of strategies for assembly. So we use 5 different assemblers, different scaffolders, different method for polishing the data, looked at different quality metrics, and we subsetted the data by read length, between 5 KB, 10 KB, 22 KB, and reads larger than 30 KB. So we did this, basically ended up with 144 genome assemblies using all those different approaches. And this work was published by Anne in Molecular Ecology Resource last year. So really, a few conclusions came out from it. Is that the first round of polishing that you would do with software like Racon or Metadata would show very good improvement in the first round of polishing, and the improvement would be in terms of your N50 metrics for your contigs, as well as the total length of the assembly. It was closer to what we were expecting. But the additional round of polishing that we did when we increased the number of either Racon polishing or combination of Racon and Metadata, we found that it was actually a marginal increase after a while. You just don't need to actually continue polishing rounds after rounds. Another observation is that post-contig assembly, you can do some polishing using high-quality short-read Illumina data on your Oxford Nanopore-based contigs, and we observed that some pylon-based polishing increased quite well the gene completeness. When you look at Busco scores, they were increased when we did the polishing post contig assembly. So we looked at performance of those assembly software against each other, and we found that a software like Shasta is quite fast and accurate in terms of its completeness and the total length of the assembly and the N50 it was generating, but it performs better with the subsets that we had with higher depth. It needs quite a high sequence depth. Conversely, Canu is quite slower, and it needs longer reads. So it means that our subsets of reads with longer reads were actually performing quite much better than when we include all the data, including the shorter types of reads. And we found that the fly assembler in our hand for the Oxford Nanopore data of such type of moderately heterozygous individual was actually quite robust and gave us some pretty complete assemblies. And that's actually the one that we ended up using as the final assembly for our scaffolding. So yeah, that's a few conclusions, from Oxford Nanopore. So you can actually get sufficient amount of data to assemble most plant genomes. It's relatively inexpensive. The example of the Apple sequencing data we generated was costing us less than $4,000. It's a decent sum of money, but think about how much it cost us like 12 years ago when we put the first Apple genome together. It was a massive cost to produce the data. Some of the instruments for Oxford Nanopore are portable. Most lab can have one, but also you can imagine that some of those mini-ion sequences could be field-based. And the attractiveness is that ultra-long reads can be achieved. if you've got someone in the lab who's very capable of producing some very, very high quality and high molecular weight DNA. And the good thing is that the community from Oxford Nanopore is very active. So there's, new base calling algorithms improved all the time, new assemblers coming all the time, and methods for reagents are improving all the time also. And I'll talk about that a little bit later. Now, some issues with Oxford Nanopore is that, you can be good at extracting your DNA and have very large fragment. It looks good on your agarose gel, but sometimes you can have DNA that has contaminants that can clog the pores, and basically your experiment is not very useful. Also, the error rate for Oxford Nanopore can be quite high, and you can have values of Fred scores around Q10 or Q15, which is still quite a lot of errors in your sequence. So that means that, as I said, with the example of the rewarewa, where we did like polishing and various options for cleaning the data, that it's using those Oxford Nanopore data can create some very good genome assemblies, but it's very time consuming. Very CPU intensive, which means you use more computers, more energy, and more carbon pumped into the atmosphere. So consider that when you put those genomes together. Don't do what we did actually of putting 144 genomes together. That was informative, but it's probably overkill. Also, the thing is that in terms of polishing, is that you got to be careful that when the error rate is much higher than your heterozygosity, you've got a chance that you might actually polish, some of your variants, and that you actually might drift from the biological truth. So a little bit careful with putting too many rounds of polishing using the Oxford Nanopore data. All right, so the alternative of Oxford Nanopore is using Pacbio high fidelity technology, and again, it's creating it generates long reads. Typically, one smart cell would give you about more than 20 gigabases of data, more if you've got a very good service provider. But what's quite interesting is that the hifi technology has really improved the accuracy of base of the bases, and you get typically Fred score greater than Q30, which means that you get less than one mistake every 1,000 base pair. And in this case, that means that for some species where you've got several SNPs or variants per kilobase, it means that your error rate is suddenly less big than your heterozygosity, which is interesting. So it changes a little. It shifts everything in terms of your error correction and haplotyping and so on. But I'll talk about it. So I'll show a couple of examples that we've done with two native species of New Zealand. One is called swamp maire. It's part of the Myrtaceae family, and the other one is Karaka. It's part of the Cucurbitaceae order. So I'll start with swamp maire, which we've done with my PhD student Colin Balkwill and a friend from the local Maori community, Paul Horton. And what we did is that, so it's quite a compact genome. It's diploid, fortunately, 300 megabase. So a good genome to start, playing with that sort of data, not too big, and a good one to have, like something of decent size to play with. So we generated about 49x coverage of hifi data. It was about 20 gigabase of data. A very good score of Q29 from the median quality of the reads, and the N50 was a bit less than 20 kb. So what we did is that we tested 3 different assemblers. We tested Fly, Canoe, and hifi ASM. And this is the stats what we got. And what you can see is that first I didn't purge the haplotypes from Canoe, but we obtained quite different config length distribution between the 3 software. We had many more contigs produced with Canoe, with quite a good N50, and half of the assembly was contained in 19 contigs, which is pretty good. And the Busco completeness was 99%. So quite good results with Canoe. Fly also was not too bad, although more fragmented. The N50 was smaller, but we had less contigs, and the Busco score was still very, very good. Now, hifi ASM ran much faster than the other two, and the results were quite spectacular. We had a smaller number of total contigs, and we had an N50 that was, way bigger than the other two software, larger than 23 megabase, sorry, and half of the assembly was contained in only 6 contigs. And what's really, really exciting is that 4 of these contigs have, evidence of telomeric repeats at both ends. So we basically have 4 contigs that are, full chromosomes from one end to the other without gaps, without, so it's, and it's not scaffolds, it's contigs. So, and again, the Busco score was quite good. So very, very exciting results for this species. So we looked at another species called Caraca. This one is a species that's also native to New Zealand. We expect 22 chromosome, and we produced some hifi data recently. The read quality was even better, Q31 median quality. The N50 was similar to what we got with the previous example, a bit less than 20 kb. Same read depth, 47x coverage. And in this case, the smart cell produced more than 30 gigabases of data, so it was quite exciting. We did camera analysis and observed that it's not a very heterozygous species. Okay, so that's interesting. And what we did is that, well, this time I didn't bother using Canoe and Fly. I went straight to hifi ASM. And here are the statistics that I got from the assembly. So hifi ASM produces, primary and 2 haplotyped assemblies. And if you look at the statistics, first, the total size assembled for the primary and the 2 haplotypes are extremely close to what we were expecting based on flow cytometry. So it's really good news. Second, the N50 was again quite spectacular, greater, like that 27 kb. And again, the L50, very few contigs, covering more than half of the genome. The Busco scores were quite high, and interestingly, quite complete Buscos for haplotype 1 and 2. The haplotype 1 and 2 had very, very similar statistics, which is quite interesting. This assembly ran, under 4 hours, no polishing, no scaffolding. I used default parameters, and that's pretty impressive results. Now, I told you we're expecting 22 chromosomes, so 44 telomeres. Found them in all those contigs, all 44 of them, and very excitingly, out of the 22 chromosomes, 16 chromosomes have been resolved, from telomere to telomere into contigs. So extremely complete assembly. And, from the statistics that we get from haplotype 1 and 2, I'm pretty confident that we've resolved both haplotypes for this species. So just, a little conclusion about the hifi sequencing technologies that, that excellent read accuracy is a bit of a game changer, because suddenly, as I said, you've got the error rate that's actually lower than your heterozygosity. With Fred scores greater than Q30, suddenly, that changes everything. It's also very fast. It means that, the process of what we did, with a rewarewa genome with Oxford Nanopore, of having several rounds of polishing pre and post, contig assembly, we don't need to do that anymore. So much smoother on CPU usage or GPU usage. Also, it's extremely contiguous, and we've got Busco scores, that are absolutely excellent. So that's very exciting. We also generated some very complete haplotype assembly, as I showed you for the Caraca haplotype 1 and 2. So that leads me to think, for those diploid species with relatively compact genome, that's, we've done it. But what happens now with, genomes that are actually autopolyploid? can we actually assemble the likes of tetraploid, blueberry, and kiwifruit? And we're testing that at the moment. And also, can we start also assembling larger genome with this technology? And we're looking at sequencing a species of salmon that's got a genome larger than 2 gigabases. So it's in the process of generating the data right now, and I hope it's going to generate some good results. So now some issues is that you need to generate some large quantity of high quality DNA as an input for Pacbio, but that's not major issue, and you can't produce ultra long reads with this technology. But again, this is based on the results that we got, in terms of completeness and telomere to telomere assembly. This might not be an issue in most cases. All right, now I'm going back to Oxford Nanopore because I mentioned that base accuracy is actually quite important. And well, just a couple of weeks or 3 weeks ago, with my colleague Iggy, we received some data from Tetraploid blueberry, and that we sequence using the new Q20 technology from Oxford Nanopore. And this Q20 technology allows you to use duplex reads to actually increase the accuracy of base calling, and it combines, the base calling from both strands of the same DNA, single DNA molecule. And it's claiming to achieve, more than Q20 Fred score, which is 99% accuracy. So I'm showing here, the sort of results that we got using, GAPI version 6. And actually, we got a small minority of duplex reads, only 9.3%. And interestingly, some of them achieved, Q values, around 30. So quite high quality base calling. This is here another way of showing data where in the x-axis you've got the length of your read, and the N50 was about 20 kb if I remember well, whereas against the Fred quality score, and you can see that we are pushing. You've got I put a line at a Q20, and there are a decent amount of reads that are above that line. So it means like there's probably a bit more work to do to push the data more over that line, but still, it's now capable. This technology is now capable of developing and sequencing some very good quality data. So that tetraploid blueberry assembly using the Q20 technology, we put it into the Fly software and ask it to run a haplotype assembly. And what we obtain is an assembly that's about 4 times the expected genome size of blueberry, and still in many contigs, and the same distribution of contig length as what I showed you before with swarm maire. But the N50 that we obtain is starting to be decent, about half a megabase N50, and the total assembly of 4 haplotypes is contained into 845 contigs. And the BUSCO scores are starting to look really good. And interestingly, the duplicated BUSCOs are quite high, which is what we expect from a tetraploid species. All right, so my conclusions about this is that, the long read sequencing technologies like Pacbio, Oxford Nanopore, they can generate those high quality genome. They're very contiguous, showed you, we can achieve haplotypes and have very high completeness. Now, we need to continue testing those new technologies because there's new chemistry coming all the time for both, options, and there's new bioinformatics tools coming every day. So, some of the questions we may ask shortly is, can we actually increase the percentage of duplex reads in that Oxford Nanopore Q20 technology? 9.3% of duplex reads in our blueberry dataset. We probably can do better, but how can we do that? Also, can we integrate other data types, in those long read sequencing technologies, like integrating, for example, hifi, like hifi ASM, for example, is capable of integrating hifi data into the assembly process, which means that you will end up with a full genome very, very rapidly with that sort of approach, and that needs to be tested in our plant species. Also, can we use, trios or quartets, where you've got both parents and some offspring sequenced, using long reads to actually help with the haplotyping. So this is something that needs to be tested. And obviously, like we've done it in quite compact genomes of diploid species, including Caraca, that's relatively not extremely diverse. The heterozygosity wasn't that great. So can we do that in higher ploidy species? And can we tackle larger genome? Theoretically, if you've got higher ploidies of diverse species, like I mentioned, with potato, for example, it's got lots of variants per base pair. We may be able actually to really resolve those haplotypes using those high accuracy reads. Now, the two examples I showed you for three examples for benchmarking, those long reads technologies are genomes that are important to New Zealand Maori, the indigenous people of New Zealand. And that's my segue to the second half of my talk about how to do the work, how to do genomics with indigenous people. And I'll start with a very provocative statement: is that genomics has failed to deliver its promises to indigenous communities. I'll give an example: is that if you look at human genetics and the various GWAS studies that have been done on various traits and conditions, indigenous communities have been underrepresented and or under-consented for also access to their data and metadata or samples. And that led to, the success of genomics for, precision medicine haven't actually realized the potential for those indigenous community. So indigenous people are concerned about that and have a bit of lack of trust to us as a research scientist. So we need to, our community, our research science community and the genomics community needs to find new ways of doing research. Now, some important thing when you do research with indigenous people, whether it's genomics or other aspects of biology, is to take a few step backs and reflect about, why am I doing this research? If you're doing just this to be able to attract some research funding or publish a couple of more papers, that's not probably enough. You need probably deeper, better reasons to do that. And some key questions, very simple questions that you can ask, is that, what's going to be the impact of the research I'm going to do on, for example, an 8-year-old indigenous kid that lives in a very remote community in a rural town. What does it mean for this kid, And that's the sort of question you need to ask before you actually embark in those research. All right, so I'll start with a couple of slides about key documents. The first one is, the United Nations Declaration on the Rights of Indigenous People, which talks about, their right to maintain, protect heritage, traditional knowledge, and protect, also the knowledge and the property of their fauna and flora. Flora is important for us as plant biologists. So this is a key aspect that leads into, how we do research for indigenous communities and using, native species important to them. Another key document is the Nagoya Protocol, which follows from the United Nations Declaration. And that protocol is about access and benefit sharing and traditional knowledge. And it's really about, ensuring that and describing what is fair and equitable sharing of benefits from using genetic resource. And by talking about genetic resource, this is not about only, plant material, seeds and germplasm and so on. This is also all the digital sequencing data that we develop from genomic research. So there's a few, there's some information about what benefits sharing means in the Nagoya Protocol. I'll just have a look in the annex, but I'll show a few examples later of what we've done with indigenous people in New Zealand. But some examples are your monetary benefits, like access fee for collecting sample, royalties if you're doing breeding, being part of research program and receiving some funding to do research. And if you've got commercial development of your research, joint ownership of the IP or joint ventures. And there are also a number of non-monetary type of benefit sharing, and that could be, like very, very, very common sense, like sharing the research and that you are doing, as much as possible with the community way before you go into the stage of publication. Collaborate and have Indigenous people involved in project, and that often means education and training is required, but also some other like important aspects about social recognition of Indigenous people in those research, contributing to the local economy, and also helping those communities that often have a strong interest in conserving biodiversity, having bringing them that scientific information that actually helps them achieve those goals. So that's just examples. It's not exhaustive list, but it's just example of how that can be done. In the situation in New Zealand is that we've got a treaty that was signed between the Queen of England and Maori chief in 1840. And that treaty recognized that Maori have a right of guardianship and sovereignty on the treasure species, all the treasures that they are responsible for. So the term for it is taonga, for treasures, and tino rangatiratanga is about recognizing absolute sovereignty of Maori people over those treasures. And that's a key document. It's a founding document of New Zealand. And in the second article, that's clearly stated that Maori are sovereign and have the rights of guardianship over those treasures. And that means those treasures can be DNA sequencing information that we generate from those species. So what needs to be done is you need to very early in the process of not even project operation, but actually project design, to engage with your Indigenous partners, and that's what we do in partnership with Maori. And you need to discuss very early on the very hard topics, and that could be like what's going to happen during the project, what are the protocols we're going to put in place, where are we going to go, and who we're going to meet, and so on. But also how we're going to publish things, how we're going to manage all the IP and data after the project is finished, and how we're going to commercialize that, and again share the benefits and so on. That needs to happen way early in the process. Also, what's really important is that you need to have all the research, all the steps in the protocols fully informed and consented, and that's through the entire project life cycle. So again, what happens before the project, during and after the project, once your data is generated, published, and so on, what's going to happen to these data in perpetuality. So that's quite important to have this discussion, and you need to have this discussion into an environment that's culturally safe, to accept the cultural difference and worldviews, and being able to present often conflicting views together and agree on the outcome and how to work together. So I'll show some example of some work that we've done. This is here what we call a genomics data life cycle, which we've developed in collaboration with some colleagues that wasn't working on plant genomes. I was working on fish genomes, and I was with Maori communities. And they asked us to show us what's the life cycle of the data sets, and from like the top one, step number one, wananga, which means meeting the original meeting with Maori, to the very last steps of what do we do in terms of reuse of the data. And for each of those steps, we set up a bunch of questions that are critical to answer, and what are the actions that we need to do. Another example of tools that we put in place is when we sequenced another species that's native to New Zealand. It's called Manuka. It's another Myrtaceae. So we published that genome in 2019. And what we did is that we didn't deposit this genome assembly into NCBI or any other database. We put it into a data repository that's hosted by Genomics Aotearoa. It's physically located in New Zealand, and what's happening is that if researchers overseas and or everywhere in the world want to use the Manuka genome, they can have access to it, but they need to go through a very short process of asking permission from the indigenous guardians of this data set. So a little bit few more steps than a couple of clicks to get access to a genome, but still doable and respectful of the guardianship of this genome by indigenous people. Another initiative led by my colleagues, Jane Anderson and Maui Hudson, is to actually generate or create some metadata fields that stick to your DNA data in database perpetually, and that attributes those DNA data sets to their rightful indigenous communities. And what's happening there is that a few labels, they call them biocultural labels, are developed, and those labels, those metadata fields can tell you, what's the provenance, whether or not it's been consented, if the indigenous communities are open to collaboration or commercialization or no commercialization at all. So that is associated as any other metadata field that you would find in a repository like NCBI, for example. You would have those biocultural labels that clearly attributes the data to their indigenous community. And we've used that for the first time. This is not a New Zealand example. We actually sequenced the bilberry genome a couple of years ago, and that bilberry sample we used for sequencing was from Norway, which is in a region that's in the ancestral land of the Sámi people. And what we did is that when we put it into the genomic database for Vaccinium, we put a biocultural notice to say that, to recognize that this data set is associated with these people, and showing how we intend to reuse these data set in the future. All right, another key aspect of working with Maori for doing genomics is to have Maori being involved, embedded in the research, from the co-design to publishing and so on. So I'll just show the example of Manuka, which is Manuka is a bush in New Zealand that's the basis of a high quality honey industry. It's a booming industry, and it's a native species with very high potential for very high quality honey. And most of the Manuka grows on Maori-owned land. So what some of our Maori partners who have, agribusinesses in the honey industry in New Zealand ask us to do is to look at the genetic diversity across the entire genetic genome of Manuka and distribution of the species, and looked at whether or not there was different provenances of Manuka. And yes, we found different provenances. And what it led to is actually to be able to commercialize the honey based on its, indigenous provenance. So very, very exciting for them to not only reconnect the species growing on the ancestral land to their communities, but also to use that in a commercial, for commercial benefit. So that's what we've done in terms of commercial benefit, but also what we did is that when we published this population genomics study, is that we had Maori involved as co-authors. So that's another way of doing benefit sharing by co-authorship. And in this case, I highlighted the 3 co-authors who have Maori ancestry. One of them did data analysis and bioinformatics analysis. Another one did the DNA extraction. Another one was coordinating all the sampling in the field, and the last one was co-designing the project, telling us this is how we need to operate, this is what the goal of the project is, and so on. So this is an example here of doing research for Maori with Maori. Project developed by Maori agribusinesses, answering the questions that they had. There was also a Maori governance group for the project. The project's not finished, but we had a group of Maori that were actually governing the way we were doing the engagement and managing the data. And there were 2 Maori students, grad students, trained in this project. So we built some capability also along the way. And this data set that we generated was full genome resequencing of many Manuka plants across its natural distribution. We wanted to reuse this data for another purpose, for looking at loci linked to myrtle rust resistance. So it's myrtle rust is a tree-killing fungi that's affecting Manuka. And in this case, even if the data was stored in our servers, we had to re-engage and reconsent for reuse of this data. And they accepted it. And now we're working on this particular project at the moment. This is another example. I mentioned rewarewa, that native tree. It's also the basis of another honey industry in New Zealand, and we've done the same. We've done population genomics to look at the diversity of that species in its distribution in New Zealand. One last example is, of doing research with Maori is, that species swamp maire that we assemble with the hifi Pacbio data. Is that it's part of the genus Syzygium, which is quite a large genus, but it's the only native endemic species of that genus in New Zealand, and it's highly endangered, very fragmented habitats, and very susceptible to the myrtle rust fungus. So our local Maori iwi, the local tribe, approached us and say, well, what can we do about this tree? We've got to replant quite a few of those trees to conserve them. How can we choose the best trees to replant? How can we conserve the diversity for the future? So that's, we say, well, we probably can use a few genomics tools to do that. So we generated a high quality genome, and we've also generated whole genome resequencing for more than 200 specimens, which we're analyzing at the moment. All right, so I'll just briefly talk about building capability and capacity, which is very critical, because often researchers, we want to engage with indigenous communities, but often those indigenous communities and their leaders have very limited amount of time or understanding of our research, all the intricacy of what we do. So this is our job also, to build that capability and capacity. And if it doesn't exist in those communities, we need to support it. So they can, you can have someone recognized by, paying them for their time if they help you with this engagement and so on. But other aspect that you can do is, training the next generation of researchers. There's a very low representation of indigenous people in our workforce in the research community. So we need to build to develop those new people for the future and just reestablish the balance. And one initiative from some colleagues, including Maui Hudson, which I mentioned earlier, is the Summer Institute of Indigenous Genomics, which started in North America with First Nations over there, and 6 years ago was applied to New Zealand, Aotearoa, and with some very successful workshops. There was an international conference in 2020, and a couple of years ago there was a Sing Australia. So that allows, young Maori indigenous interns, about 20 of them every year, to go into a very intensive, full week of training about, not only technical, but also ethical aspects of genomics. All right, so my conclusions, very few basic points about, common sense, engage very early with communities and obtain those informed consent to do research about indigenous species. Share benefits with those communities, and I showed a few examples how to do that. Build capability and capacity, contribute to that to help their communities. And as Western scientists, European or any other ethnicity, we need to respect indigenous people worldview, their cultural needs, and also protect their traditional knowledge. And we do that by developing culturally safe environments for discussions. And I show that there's a few tools and protocols that can be put in place to actually protect their sovereignty over this data, whether it's biocultural labels for storing metadata, or storing the raw and analyzed data into managed repositories, or developing some protocols to follow, every step of your research project to see what are the key indigenous consideration and answer some key questions. All right, I've got a few photos here of, I was very fortunate to go around the country in New Zealand, collect some various different tree species. It was a very, and most importantly, meet some wonderful people, which is very important. We spend a lot of time in front of computers assembling genomes and analyzing very large data sets, but it's a wonderful experience to go and meet the people in the field and understand, their needs, their aspirations firsthand. Very humbling and very enriching. All right, so I've got quite a lot of people to thank, from Plant and Food Research, Genomics Aotearoa, and all the partners in Genomics Aotearoa. I've listed them here. I'd like also to thank the Maori partners, all the people we've been interacting with for those projects. It was a privilege to get access to their treasures for this research, and I thank them for that. And my last slide is a couple of things, like got my contact details, but also we've got some vacancies, we've got some jobs available in our group at the moment, especially bioinformatics jobs. So have a look at the link that's on that slide, and yeah, just apply if you're interested. And I thank you very much for your attention, and I'll take questions.